Okay, for those at UC Davis, I'm Sarjeet Gill. I'm in the Department of Molecular Cell and Systems Biology at UC Riverside. And this talk I'm going to give is going to be a very general one, mostly because the audience is also probably would require a bit more general talk. Now, the work here is primarily been supported by NIH, National Institutes of Health, with other support from the Ag Experiment Station, and also from the CDC through the Pacific Southwest Vector-Borne Diseases Center. So I'm going to provide an outline, which first would be giving you an introduction, then talk about some of the receptors that are involved in toxin action, then talk about some of the roles of multiple toxins acting, that is the cry and cytotoxins, and then a general mode of action with some general conclusions. Now then, a number of bacterial toxins or bacterial species that have bacterial toxins. Now the two main ones that are involved in vector control right now are Bacillus thuringiensis and Licetes sphericus. And there are other bacterial species, for example, from Photorhabdus and Xenorhabdus, which are active on Lepidopterans. Clostridium, which we are also working on, which is active on Anopheles species, and a few others listed on the right. Now, the main topic I'm going to talk about is essentially from thuringiensis, and the protein focus is on the cryoproteins, which affect many insect orders, including those on dipterans, and the site proteins, which are primarily active against dipterans, although they can be active on other species as such. They are also, thuringiensis also produces binary proteins, and which are active against Lepidopterans and Coleopterans, and vegetative insect proteins which are active on Lepidopterans and Coleopterans. The other one is Sphericus, which is very effective against Culex species and also Anopheles. It produces binary toxins and also another toxins, which is the MTX toxin, which is intracellular acting. Now, in most of the toxins that are produced, like these are the toxin phylogeny, and there are many, as you can see on the top, that is the cry-1 toxins, which are active on Lepidopterans. In the middle, you have Coleopteran active, and at the bottom is the dipteran active, which is active against mosquitoes. The reason we are interested is because if you look at the bottom, is because of mosquitoes, which are vector-borne, which are transmit vector-borne diseases. Okay. So I'm just going to give you a brief outline of the general mechanism of action of cryotoxins in general. So what happens if you start from the top, you get spore and crystals, and these are ingested by the insect. Crystals generally get solubilized, and they get activated. And this process, there is one where there is some resistance in protea, which is due to an increased protease activity in the gut. But the principal mechanisms of resistance that have occurred has been in terms of binding to the receptor, that is the first receptor that's involved. And there are many of these that have been identified. For example, in Sphericus, it is an alpha-glucosidase, and resistance to this is relatively easy to develop, primarily because it is a single gene is involved. And you also see in the case of BT cry1 and cry2, this also usually resistance has been quite simple, mostly primarily because there are single genes involved in this particular case. The number of proteins that have been identified at this stage are the archidine molecules and ABC transporters, and also, for example, alkaline phosphatases in this case. There is also, for example, in C. elegans, further down you see resistance to cry4-5b, but particularly in this case in terms of lipid molecules, which are lipid mediators of resistance in this case. There is also a possibility that this also occurs in Lepidopteran insects. And so what happens in this particular case is after processing, there is a second receptor involved. I'll come into that in the mode of action. And that leads to insertion of the toxin in lipid grafts, which then subsequently forms a pore and ultimately goes to intracellular targets. Now, intracellular targets were a question mark earlier, but the resistance that has been found in Armadillo against tetraspanin, for example, which is an intracellular protein, suggests, in fact, and this is a dominant resistance, so in fact suggests that intracellular targets are important in the total overall mechanism of action of the toxin. Now, there are two main strains that are used for mosquito control. The top one, for example, is israelensis. And israelensis has been used extensively, for example, for black fly control in West Africa. And this was done in a regional level in the Niger Delta. It was for the control of black fly for onchocerciasis control, which is triple blindness. So BTI, together with the use of ivermectin, led to essentially the attenuation of the disease in that particular area. Now, BTI also has been used in the Rhine Valley primarily for control of nuisance mosquitoes, and to a lesser extent in control programs in California and Florida, and also, for example, in Southeast Asia. Now, one of the features is that in spite of many years of use, there has been no resistance observed in this particular case in BTI. I'll come to an explanation as to why this occurs. The next species is Lycinia sphericus, and this has been used primarily for QLEX control. Resistance has been quite rapid in Brazil, Europe, and India and China, for example. Resistance is primarily due to a single gene target, that is in alpha-glucosidase. Now, if you look at the mosquitocidal activity of those species, Aedes is particularly active against Aedes and QLEX. Israelensis is particularly active against Aedes and QLEX, and also, for example, in Anopheles. And Sphericus has a slightly different spectrum, as indicated on the page, on the slide. And the Paraclostridium, which I'm working on, has a slightly different spectrum of activity. And I'm not going to talk about that at all today. And that has activity against Anopheles species. Now, the reason we tend to work with this bacteria is because of the species of the diseases that these mosquitoes transmit. QLEX transmits West Nile virus and also filariasis. So the control of this, particularly in Brazil, for example, was quite important. And also, for example, they've used in control in Europe and in China, for example. Now, thuringiensis has not been extensively used for control of Aedes, although this mosquito is quite important for dengue and yellow fever. Now, the last one, the reason I'm interested in is Paraclostridium is because this is toxic to Anopheles, which is a vector of malaria, but I'm not going to go into much of this anymore. Now, to focus on BTI in particular, now BTI, as you can see, has an inclusion, as you can see on the top right. It has toxins, which are four main toxins involved. And you see on the table on the left that you have inclusion, that's BTI inclusion, which includes at least 6 different proteins. And 4 of them are listed here. That's Tri4A, 4B, 11A, and Site A. And if you see on the right-hand side at the bottom, and that is the STS page, which shows you the approximate size of the molecules that are involved in here. Tri4A and 4B are large proteins, they're around 130 kilodalton. 11A is about 70 kilodalton. And the Site A protein, which is the cytolytic protein, is 27 kilodalton. One of the features that is important in this particular case, go back to the table, you see on the top, the BTI inclusion itself, if you just look at the table for Aedes, it has a toxicity of about 4 nanograms per mil. But if you look at each of the individual proteins, 4A, 4B, 11A, and site A, none of them have the activity that is together, present in the inclusion. So all of these toxins collectively contribute and also have synergistic activity that contributes to increased activity of the inclusion itself. So one of the features that is known, that has been done, which Bride has done a lot also, is that the cry and cytotoxins have a synergistic activity, and they play an important role in the overall toxicity of this particular strain. So the mechanism, crudely, is that it acts on the microvilli on the left-hand side, and those are the microvilli on an untreated insect, and if you treat shortly, it gets, the microvilli are destroyed and basically leads to cell lysis that occurs. Now, BTI, all the 4 toxins that are produced are encoded in a large plasmid of approximately 128 kilobase. And it has, as you can see in the middle of the page, it's called PB-toxis. And it includes, as you can see, there are 7 different genes involved. But the principal ones which are active are the ones I showed earlier. That is 4A, 4B, 11A, and site 1A. I'm working primarily on the Cry11A and Cry4B and the site 1A toxin. So the rest of the talk primarily is going to focus on these 2 proteins as such. So one of the features that is important for understanding the mechanism of these toxins is that the site toxins are a 3-domain structure. And if you look at the figure, the top figure on the left-hand side, which is a structure, the blue or the cyan structure are alpha helices. And these alpha helices are the pore-forming component of the toxin. The green structure in that particular one is the receptor binding domain. And then you have the purple one that is domain 3. It actually modulates both the binding and also the pore formation. So this is a 3-domain type of structure. And then that is quite distinctly different from the site toxin structure, which is at the bottom right. In this case, you have the alpha helices covering up basically the beta sheets. And the way these toxins act are slightly different. The way they form pores are different. The 3-domain toxins form an alpha helical structure pore, where the pore that includes basically the alpha helical structures. This primarily includes helix 4 and 5 from that goes into the membrane. Whereas in case of the site toxin, it is a beta barrel. So the beta sheets essentially form a beta barrel pore. So the toxins have very different mechanisms of action, and they are both encoded by the same bacteria. So another component that is important in understanding the mechanism of the toxin is these toxins, especially in domain 2, has unique loop regions. And the loop regions are indicated in this diagram, where the red is the loop 1, the purple is loop 2, the yellow is loop 3, and the cyan is in loop 4. In particular for Cry11A, the loop alpha 8 is quite important, so keep that in mind when I'm going to talk about that. Also, loop 3 also plays a role in the case of alpha Cry11A toxicity. So to understand which of the loops are involved, the way that investigators and have studied, and this was work that was done collaboratively with the Mexican group in my lab, was that you can make peptide regions to each of the loop regions and then determine whether they compete with the binding of the toxin to the brush border membrane of mosquitoes. So that is a very simple way of studying. So if you want to study which of the loops are involved or which domains are involved, you make peptides against different regions. and then you can basically use it in a competition assay. And in this particular case, you can see, if you look at the top figure, the beta sheet 4A and 4B do compete, and also loop alpha 8 in the second panel also compete, and loop alpha 3 also competes. And loop alpha 8 is particularly more important than the other two. So one of the features that you can see is where Cry11A binds to the midcut. Now, just focus your attention on the top row of panels. The Cry11A in the top row on the left-hand panel, it binds to the brush border membrane, and this is the apical cells of the posterior gut that binds. And if you do this binding and compete that with loop alpha 8 peptides, for example, you see that you essentially have completed nearly most of the binding that is present. And in this way, we can identify which are the domains and which of the amino acids are involved in actually binding to the midgut. Now, as you can see further, loop beta 4 and amide beta 4 sheet and loop 3 are not as well, do not compete as well as loop alpha 8. Now, once you have done that, and that's what we did, we then can go further and try to identify specific amino acids which are involved in that particular domain which are involved in binding. So then that domain has about 6 or 7 amino acids, and we did basically mutation of each of the amino acids, replace them with alanine, and then determine whether it can also bind. What you will get is essentially an experiment that you see, for example, like this on the graph on the right. And the graph on the right, for example, the line in red is where we have mutated in loop alpha 8 the glutamic acid substituted with alanine. And you can see it competes much less compared to the wild type toxin, which is at the bottom, the most bottom curve that is present. So in this way, we can identify specific amino acids that are involved. And in this case, the amino acid that is involved is, this is the amino acid that's involved in binding a specific protein, that is the adhering protein in this case. So if you look at this, so the two amino acids that are critical in this case is the glutamic acid 266 and the valine 262, for example. So if you mutate the glutamic acid with alanine, and on the right-hand side, you can see that it is unable to compete. The top line indicates it's unable to compete, whereas the 262 mutation is still able to compete, showing that the valine 262 is not as important as the 266 amino acid in this particular case. Just to indicate that we have another receptor which we have not published on, it is another cadherin that binds a totally different, it actually, in that particular case, the valine 262 is more important than the glutamic acid in this particular case. So, I've been talking about essentially the toxin as such. Now, I want to flip on the other side of the partner that's involved. So you have in the midgut, you have the toxin binding to receptor. So what are the receptors involved in this particular case? So you can do this in pull-down experiments or membrane fraction. And if you do, depending on how you do a pull-down, if you do a pull-down with a GPI anchor cleave, you see slightly different proteins that are obtained. But overall, we can get three main groups of proteins that are identified. One is around 250 kilodalton, the other one around 100, and the other next one is around 65 and 62 kilodalton. The two smaller proteins are both GPI anchored proteins. That means they have a GPI anchored to the protein. And the 100 kilodalton protein is an aminopeptidase. The 65 kilodalton protein is an alkaline phosphatase. The 250 kilodalton protein is an cadherin. We've characterized all three proteins and identified which they are. So based on this structure, based on the proteins that are actually identified here, especially the large protein, we can basically summarize that the mechanism of this particular toxin is similar to that of other lepidopteran toxins identified. So to prove which of the protein, that the 250 kilodalton protein is essentially a receptor, what we did was we purified a partial fragment of this protein, develop an antibody to that, and use an antibody in a competition assay. Previously, we used a peptide. Now, in this case, we use a peptide, an antibody to a fraction of the protein, and then do a competition assay. And on the panel on the right, you can see in lane 1, essentially is where the toxin is able to bind. It binds in high amount to the brush border membranes of the mosquito. And if you add antibody, in this case, 1 to 1,000 dilution in lane 2, and lane 3 is 1 to 1,000 dilution. The lanes at the bottom are actually misplaced a bit. So you can see that the antibody is able to compete against the binding of the toxin, suggesting that this particular protein is important in binding, and the antibody is competing with this. Once we got this data, we then basically cloned the whole protein, and this is a large protein. It's 250 kilodalton, so it took some time for cloning. And this was before the days of the genome of the mosquito was known, so it was a bit more challenging. So what you see is this protein has what is called cadherin repeats. There are 11 of them, and these are generally thought to be involved in some aspects of the binding. So if we analyze the binding of this protein, the way we actually determine which part of this protein is involved in binding to a toxin, the way we do that is we make partial fragments which are above the shown above the cadherin repeats. You have repeat 1 and 2, 1 to 5, 3 to 7, and 7 to 11. That is, we make these domains and use them to compete against binding. Having done that, what we found that the cadherin repeats from 7 to 11 were the ones where the toxin actually binds. We have subsequently determined actually is much closer to cadherin 11 as where the toxin actually binds. So if you look at that and look at the panel on the right, the graph, you can see only one of the domains is actually binding, that is cadherin repeat 7 to 11 is the only one that is able to bind. So that region is quite important. So where is cadherin itself expressed? These are recent experiments which Genwu did. And in this case, what we have done is we have labeled the cadherin protein itself in vivo by a fusion construct and show where it is indeed expressed. So if you look at the top figure, the top row of figures, top panel, in the middle panel is where the protein which is tagged by EGFP, you can see it is expressed on the CK, which is on the left, and the posterior gut, which is on the right. In between the two is what is the anterior gut. In mosquito, the anterior gut is very different in pH. It is alkaline, whereas the posterior gut is neutral, and so is the CK. So the toxin actually binds to the cadherin is expressed in the CK and in the posterior gut. And in the middle panel, you can see in the middle that is expressed in the CK, in the epithelial cells. And in the bottom panel, you can see it's expressed in the apical cells. So what you're seeing is that the cadherin is expressed in only certain parts. Now, to determine whether the Cry11A toxin actually binds to this region, we then label the Cry11A toxin with Alexa, and then co-incubated this, and you can see the panel in the mid, cadherin is in green. You see it binds. That's where cadherin is expressed. And then Cry11A is in red, and you can see it binds to a certain region. And when you merge the two figures, you can see essentially the right-hand figure, which is in yellow, that cry11A actually binds to most regions, or nearly all regions, which expresses the cadherin. Not entirely, but most regions, because cadherin expression is slightly in some areas where the toxin is unable to get to. So cry11A binds to the same regions that expresses the cadherin EGFP. Thus, cadherin is a likely receptor for cry11A toxin. So one of the things that we are trying to do is try to knock out this cadherin receptor in vivo, because you can do that in Lepidopterans, but we cannot do that in Aedes mosquitoes. It is unlike in Lepidopterans, this cadherin gene is essential for mosquitoes, but it's not so in the case of Lepidopterans, where you can do a CRISPR knockouts. You cannot do CRISPR knockouts in this particular case. So I've already talked about the larger protein, the 250 kilodalton protein. Then I'm going to talk about the other proteins. The second one is the 100 kilodalton, and then I'll go into the 65 kilodalton protein. The 100 kilodalton protein is an aminopeptidases. We have identified two of them. I don't want to go into much detail, just to show that they actually have relatively good affinity. They are in the nanomolar levels of affinity, so that they do play a role in the overall toxicity of the cry11A toxin. Now, we also did a similar experiment like we did in cadherin, where we developed antibodies to alkaline phosphatase. And you can see in the middle panel where the antibodies are able to compete for the binding to alkaline phosphatase. So indeed, in this particular case, it is also an important mediator of toxicity. So if based on this, just on these particular figures, on this data, then you can essentially come up with a putative model of toxicity. That is, cry11A will bind, for example, to cadherin receptor, or it can bind to a GPI-anchored protein, for example, in ALP or APN. Or the more likely scenario is the GPI-anchored protein is used to sequester the toxin, which then binds to cadherin, which is a high affinity. And then it oligomerizes. And then I have not put an intermediate step. That actually is a sequential binding. It then binds as an oligomer to the alkaline phosphatase in the lipid raft, and then inserts, and then basically forms core. So we generally thought that we have a good idea of how cry11A binds, and so we also believe that the other cry toxins in here, cry4B and cry11A, also bind in a similar mechanism. But we were surprised that, in fact, cry4B has a different mechanism than cry11A. So, because it doesn't bind to cadherin molecule, for example, cry4B doesn't bind to cadherin. So to evaluate first what Aedes cadherin does in vivo in terms of toxicity, we silence this cadherin gene in vivo using transgenic mosquitoes for RNAi silencing. We do that in vivo because when you do feeding, the data is actually quite equivocal and quite variable to a large extent. Whereas you do it in vivo, it is much stronger and the data is quite tight. So we express this, and on the right-hand panel is where with the heat shock, you will see a decrease in level of expression. That is a fold change of around 6.5. And this is with and without heat shock, and you can see this is a leaky promoter that without, this was a Drosophila heat shock promoter we used, and we have now subsequently moved to the Aedes heat shock promoter, which is actually much tighter, it doesn't leak as much. Using this mosquito, we then determine the toxicity to cry11A in the top panel on the right, and to cry4B at the bottom panel on the right. Just focus on those two. You can see that in the dark blue or black line, that is the wild-type mosquito, and in the red line is the mosquito that is in silence. Just by silencing this gene is approximately around 15-fold difference just by silencing this gene. So it is important for cry11A, but it is not important for cry4B, because silencing this gene does not attenuate or change the toxicity from cry4b. So it's a slightly different target site that occurs. So we also did binding, and you can see just focus on the panel with multiple lines, and you can see the figure in cry4b is a line on top. It does not compete, for example, with binding to cadherin as such. And if you look at the table at the bottom, if you look at the figures, cry11A has a very high affinity of around 17 nanomolar, whereas cry4b has essentially, we can measure that, but it is not of a significant value because it's more than 100 nanomolar as the data shows. So when we put this inside, and then we look at the mechanism of action that occurs, so the cry4b binds to another receptor. In this case, it looks to be binding directly to alkaline phosphatases. The reason it does is because cry4b, once it's activated, it spontaneously oligomerizes. It doesn't need cadherin molecule to bind and then oligomerize. So there's a two-stage process that occurs. So there's a slightly different process that occurs. So if a mosquito is to develop resistance to cadherin, for example, cry4b will still be functioning, and so BTI would still be toxic as such. So now I'm going to do another thing. So one of the ways we're trying to look at what are the genes, other genes that are involved in resistance mechanism. So we have developed a resistance in Aedes aegypti because all our work is in Aedes aegypti. And in this case, what we had used was basically an EMS-treated lab only and a selection of a number of years. And with the wild type toxin, we can develop much higher levels of resistance. I'll come into that. But look at the panel on the right, that the cry11A resistant strain is resistant to cry11A, less to cry4A, but is not resistant to cry4B, and surprisingly to us, to cry11B, which is a very related toxin to cry11A. So why that's the case, we are not certain. So Harlan has also developed resistance from using a purified toxin. And in the purified toxin, that's a figure on the bottom, you can see that now we have generation 60 in this case, that the wild type is on the left and the resistance strain is on the right. Again, this is against a pure purified inclusion. And this resistance fold is approximately 100, whereas to crude toxin, the resistance is approximately 1,000. So you can develop resistance to individual toxins, but you cannot develop resistance to the full strain as such. So to understand what are the genes involved, we have done an RNA-seq of the resistant strain, and this we did at generation 50, although we are now in generation 60. What you observe is, if you look at these two panels, the panel on the left is the resistant strain, the panel on the right is the susceptible strain. But you can see that there are some differences in the middle of the panel. On the top, they seem to be similar, but in the middle, there are a number of genes which are either attenuated or changed. Altogether, about 760 genes are altered, and some are high expressed, some are lower expressed. But this is trying to find a needle in a haystack. It's actually quite difficult to understand which of the genes is involved. So Harlan is actually trying to do much, probably we have to do genetic mapping to find where the gene is involved. And so that is the next stage of work that we are hoping to continue. So I'm going to come back to another toxin. I've been talking about cry11A entirely. Now I'm going to talk about to the site toxin. Now, if you look at the table itself, the site toxin is the last line, which is the toxic, which is in green. What happens if you mix this toxin with either cry11A or 4A, the toxicity of the cryotoxin increases. Now, our model is that the synergistic effect between this toxin is due to molecular interactions between the toxins. Now, and this work was done by Claudia Perez in Alejandro Bravo's lab. And so the data, I'm going to show you one particular slide of data. one slide, and you see on the left-hand panel where the BBME, which is incubated with the cytotoxin or without the cytotoxin. On the left lane, without the cytotoxin, you see that cry11A only binds a limited amount. But if you add site A, it shows there is a large amount of binding. In the second panel, if you put, for example, another toxin, which is not doesn't insert into membrane, you do not see any increase in binding. That's a control. Now, the way this occurs is if you look on the right-hand side, that there are domains that are involved in the cytotoxin, which are able to bind to specific sites on the cry11A toxin. And the sites that bind in the cry11A to site A are the same sites in cry11A that binds to the receptor. So basically what you're saying is the cytolytic protein acts as a surrogate receptor for the membrane protein. So this is an example. This is what I'm talking about. So if you look cry11A, on the left-hand side is cytotoxin. It binds to specific epitopes on the protein. And it can bind to the membrane, in that case, the receptor, which is on the membrane. In this case, for example, a cadherin protein, which is on the receptor. So therefore, the cytotoxin actually acts as an alternative receptor of the cry11A protein. So therefore, we can come with an alternative mode of action that occurs in this case. So this is a more complicated slide. So if you look, from the first part is the slide I showed you earlier. What I've added is at the bottom, the cytotoxin. So the cytotoxin inserts by itself into the membrane. What it requires is lipid molecules. Its recognition motif is unsaturated lipids that are on the cell membrane. It recognizes them and inserts into the membrane because of that. So it inserts without a receptor. But once it is inserted, it can then bind cry11A, or it can also bind cry4B because that also has an interaction that occurs. So what you have is, as an alternative, if it binds to that, it still undergoes through an oligomerization process and inserts into the membrane. So the cry11A can go, for example, in this process, in the bottom process, or the side cry11A can go into the middle process, or the cry4B can go through the top mechanism. So as a summary, basically what you see is the toxin, the BTI toxins have multiple modes of action. The cry11A has a slightly different mode of action. The cytotoxin is a different mode, and so is cry4B is different. So the synergism that occurs is between cry11A and the cytotoxin, and the cry4B and the cytotoxin. We do not know whether there's any synergism between the cry4B and the cry11A, but there are different mechanisms that occur. So if there is a resistance that would occur, for example, in Lepidopterans against the cadherin molecule, it will not develop resistance in this particular case because there are alternative modes of action and there are different ways that the toxin could occur. Now, I did not talk about one process and I just alluded to it, that is between the first binding step and the second binding step. The first binding occurs on the membrane. The second binding also occurs on the membrane, but it actually moves the receptor, cadherin receptor, to the lipid raft. And this is what we showed it in Lepidopterans. We have not showed it occurs in mosquitoes, but generally the same process should occur in this particular case. So in conclusion, what I'm going to say is basically that cry11A has multiple receptors. The cry4B has also some receptors, but they're different. Some of them could be the same. For example, ALP seems to be the same in this case. And then the cry11A can synergize the crytoxin activity primarily by acting as an alternative or receptor or as a surrogate receptor. And there are specific domains that are involved in cry11A and site A, which are involved in toxin interactions. That is, if you mutate those areas, you essentially lose these interactions that occur. Now, critically, domains that are involved in cry-11A which bind to the receptor are the same domains that are involved in binding to the site on a toxin. That is, loop alpha 8 is involved in binding to the receptor, cadherin, for example, and that same loop domain, loop alpha 8, is involved in binding to the site 1A protein. Oligomerization is an essential process. I did not go into that extensively, but this is likely process which is required for the second stage of binding process. That is, it oligomerizes and then goes into lipid rafts. Why is this important? Because the binding affinity of an oligomer is different from a monomer. The monomer has a high affinity, for example, to the cadherin molecule, whereas the oligomer has a high affinity for the GPI-anchored proteins. So this is actually quite critical as such. So it is this complexity in the toxin mechanism, there are multiple modes of action, which explains why resistance in BTI has not been observed even after 3 decades of use in the field. So finally, I would like to thank my lab members. Most of this work was done by Jen and Kali Aminova and others who are not in the lab right now. And also Arnan Jang has been continuing to do the work on the resistance. Thank you.